Identifying the target DNA sequence in CRISPR, clustered regularly interspaced short palindromic repeats technology, is a crucial step in genome editing. The process involves selecting a specific DNA sequence to be edited, modified, or knocked out. Here's how it's done. 1. Understanding the CRISPR-Cas system. CRISPR technology relies on the Cas, CRISPR-associated proteins, especially Cas9, which uses a guide RNA, gRNA, to find and bind to a specific DNA sequence. Part 2. Steps for identifying the target DNA sequence. A. Define the objective. Determine the gene of interest, e.g. disease-causing mutation, desired trait modification. Choose whether you want to knock out, knock in, or modify the gene. Part B. Locate the target sequence. The target sequence should be 20 nucleotides long and must be adjacent to a protospacer's or adjacent motif, a papiam symbiotic soin. For Cas9 from Streptococcus pyogenes SPCas9, the PAM sequence is NGG, where N is any nucleotide. Example, 5 find GAT-TCGG fresh. C. Use bioinformatics tools, genome databases, eEnsemble, NCBI, UCSC genome browser, help locate gene sequences. CRISPR design tools, e.g. ChopChop, Benchling, CRISPR Scan, CRISPR Direct, predict best gRNA sequences, specificity to avoid off-target effects. Efficiency scores, D. Check for uniqueness and specificity. The chosen sequence must be unique to avoid unintended edits. Tools like BLAST, Basic Local Alignment Search Tool, help check for off-target binding. E. Validate in silico. Computational modeling can predict potential off-target sites, efficiency of CRISPR-Cas binding, and cleavage. Experimental validation. After in silico identification, 1. Synthesize gRNA, create the RNA sequence complementary to the target DNA. 2. Test in cell cultures, confirm target recognition in vitro. 3. Sequencing analysis. Verify edits using Sanger sequencing or next-generation sequencing, NGS. Conclusion. Identifying the target DNA sequence in CRISPR involves bioinformatics analysis, specificity checks, and experimental validation to ensure precise genome editing with minimal off-target effects.